This chapter 20 is essentially the anatomy of the lymphatic system. The next chapter, chapter 21, is the physiology, immunology. How does all this stuff come together, all these cells that are inside our lymphatic system and all the systems that the lymphatic system helps? How does all that work together? Well, why is this important? Well, more often than not, we evaluate the lymph nodes for signs of infection or signs of cancer. Your doctor always is going to palpate your neck. He's always going to be feeling for your cervical lymph nodes to see if they're swollen, whether they're tender or not. So it's part of your physical exam. We've been through the function of the lymphatic system, but we'll go through it on this slide again. So the lymphatic system is housing those immune cells. The biggies are going to be the lymphocytes, but there's also going to be some macrophages and dendritic cells, which the difference, I'll probably say this again later, but a macrophage is a cell that phagocyzes bacteria. He mostly patrols connective tissue. Dendritic cells are essentially the same thing. They look a little bit more spiky, and they're patrolling more of the epithelial areas of the body. Then those cells, they're going to be housed, and they're going to learn how to fight inside a majority of the lymphatic tissue. They're going to have to mature in two particular areas before they can learn how to fight, and we're going to go through all those different things. And then finally, we've talked about how vessels, because of hydrostatic pressure, you're going to lose a little bit of fluid to the extracellular areas, but we're going to have an osmotic pull, pulling most of it back in, and then there's a small little bit left. Well, when you talk about the entire body, you actually end up pushing about 3 liters total that need to be picked up by the lymph system every day. Okay, so what does it consist of? Well, there's lots of lymph vessels, so they're going to be tiny capillaries, and then they're going to become larger type vessels, which are going to go into trunks, which are going to end up going into ducts. You don't need to know those pathways. The fluid inside of these vessels, for the most part, is called lymph. I'm going to go into one little area where we're going to change his name up a little bit. And the lymph nodes are little bean-sized areas that the lymph vessels pass through every now and then, which is the cleaning stations. Okay, so the difference between lymphatic capillaries and the circulatory pathways capillaries is that they have an end. They have a blind end. You remember from circulatory, those capillaries are going to be arterial and go to tinier and tinier and tinier capillaries, and then we're going to have a capillary bed where then it's going to become a venous side, but it never stops. In the lymphatic capillaries, they actually have a blunted end. So that's different. Another thing is they are permeable like blood capillaries, but even more because they don't have tight junctions. So I'm going to show you a picture of that in a second. They can pick up larger molecules. So if you have an infection, you're going to lose some proteins out of your vessels. So when you have a blood-borne infection, your capillaries are going to weep out excess fluid. But it's okay because your lymphatics are going to be able to pick up that excess fluid and anything that got dumped out that wasn't supposed to get dumped out. So maybe you lost too much albumin. That's okay because your lymphatics are going to help pick that up. Can also pick up other things. Glucose is a pretty big molecule. You can pick that guy back up. Any sort of debris. So if you have an infection, you probably have an increased number of lymphocytes. Maybe some of them are dying already if it's sepsis, so infection in the blood. But the lymphatic system is going to help clean those guys up. Any bacteria, things that are considered pathogens, so parasites too. And then alas, it'll also pick up cancer cells. Because lymphatics are passing all over the body, sometimes this can be a problem because that cancer cell can pass through the lymphatics of another organ and seed itself, and that's called metastasis. Okay, so there's two structures that make this guy increasingly permeable. So I mentioned one of them is the fact that he doesn't have tight junctions. Their endothelial cells overlap and they form like a one-way mini valve. This will make more sense when you see the picture. And then those mini valves are actually anchored to the tissue, the matrix around it. So when you increase the pressure because you've increased the amount of interstitial fluid, then it's going to increase the pressure on these one-way valves. It makes them stay open more because they are not going to collapse because they're anchored. Okay, this is where I was telling you, most of the time we call it lymph, but those lacteals that we mentioned in the GI section, those are specialized lymphatic capillaries in the intestinal mucosa, and they're not just lymph, but they also have some fatty acids in them. So those fats are going to mix with the lymph, and it's going to be white. So hence, just like milk has lactose, it gets its name because of its color. So chyle is what the name of the stuff is instead of lymph. It's lymph plus fatty acids. It's called chyle, and lacteals gets its name because it looks like milk. Here's that picture. So the tiniest little end, this little blunted end, the blind end of a lymphatic capillary. His cells, here's one cell, doop-de-doopy-doop, and I'm not sure you can see my pointer now that I think about it. And then there's another cell that's actually laying over it. That flap anchors to the tissues around it, the matrix around it, and it's collagen. And so if you increase the pressure on that little one-way swinging door, well, then it's going to stay open more, and the filaments help keep that vessel, that tiny little vessel, from being squished by the increase in pressure from the interstitial fluid when the interstitial fluid amount increases. So, in a nutshell, your book goes through all the different types of, they all have names, the different areas, but let's focus more on the fact that we've got some capillaries that are going to make their way into the main ducts. So, the right lymphatic duct is draining the head, mostly the right side of the head, the right side of the chest, and the right arm. Everything else is going into the thoracic duct. Both of those ducts, the right lymphatic duct and the thoracic duct, both of those guys are going to dump into the subclavian vein, kind of at the meeting of the subclavian and the internal jugular, actually. I don't have anything to say about that one. Well, sure. Okay, here's the clumping of the lymph nodes. You have lymph nodes everywhere, but these are the commonly listed ones right here. So the cervical nodes are the ones that I was talking about that the doctor usually checks on. So comes and palpates your neck. Axillary nodes, so in your armpit, you should not feel those guys. And then inguinal lymph nodes. However, you can see that there's all these little beans everywhere. And some of them are very tiny. If you look in the abdomen, the mesenteric lymph nodes are draining the areas around the gut. So if you can imagine, those guys are actually normally a little bigger. And maybe you've heard somebody say, wow, he was so sick. He got those red streaks up their arms and almost died. Well, so what's going on there? Well, if the lymphatics are picking up bad things, if it's constantly keeping areas clean, then when it's picked up, let's say you have an infection, maybe you cut your finger and you didn't clean it good, and now it's infected and throbbing, and you still don't go to the doctor. What's going to happen is the bacteria that's in your infected wound is going to start traveling up your lymphatics. Well, the lymphatic vessels, the larger ones that have left the capillaries, they actually have a vasovasorum, just like the larger vessels of the circulatory system. So that outer part has its own tiny little capillary bed that's making sure that the vessels are getting the blood supply that they need, that those endothelial cells, all the smooth muscle cells, whatever, that everybody's getting fed with nutrients. So that vasovasorum is a very tiny capillary bed, and it's inflamed. So you can actually see the vessels that are feeding the lymphatics are becoming hot and reddened because they are inflamed. They're vasodilating and they're experiencing the infection too. That's called lymphangitis. This hurts. Usually you can feel that red streak and it's kind of a burning sensation. Okay, so lymph pumping around is a low pressure system just like the venous system, and it has similar mechanisms that get the transport done. So your muscles, like in your legs, are helping pump. Your respiratory pump, so the breathing and the contraction and the movement of the organs because of your breathing. He also has valves, just like veins. And then the smallest little lymphatic capillaries, they have a smooth muscle, just like in the venous system, but they rhythmically just contract and relax, contract and relax, contract and relax. This helps transport lymph. Physical activity increases the flow of lymph. So if you're exercising, if you're running, well, then your muscles are contracting it more, so therefore it's going to increase it. So what do we do with bedridden patients to increase both lymph and venous circulation? We put those little pumps on their legs, so you wrap around their lower legs and it just stays rhythmically contracting and relaxing to help increase the circulation because pressures are so low. So hospitalized patients get the pumps around their lower legs. Okay, so anything that is going to keep lymph from getting back to the blood, you're going to end up with a buildup of lymph in the tissues. So interstitial tissue is not getting the lymphatics to the lymphatics are not picking up the interstitial fluid. So it's called lymphedema because it's not just fluid that has left the vessels. It is accumulating because the lymphatic system is not working right. And even sometimes you might even get a little bit of lymph leaking into the interstitial fluid as well. So what can cause that? Well, do you have lymphatics that are blocked for some reason? Maybe so full of debris, but more likely maybe cancer. Or maybe you've had a surgery and they've removed some of your lymphatics, and so you've lost the lymphatic pickup system. Hopefully, when you've had the lymphatics pulled, that there's some left, and those areas eventually will adjust and enlarge and help minimize the edema. But let's go with the cancer patients, like breast cancer patients. And they've checked the sentinel node and they see one in the sentinel node. The sentinel node is just the first lymph node to be draining an area. So you saw that picture of the whole chain. So in the area, there's a bunch of axillaries, there's a bunch of cervical, there's a bunch of inguinal. The first lymph node that is draining a particular area, in this case, we're talking about cancer, the first one that gets lymph is going to be the sentinel node. The lymph is going to pass through that lymph node, get clean up stuff, and then it's going to move to the next lymph node and clean up. It's like repeat, continuous cleaning. But anyway, if they take out some lymph nodes and capillaries that are associated with it, but maybe they left like the 6th lymph node and all of its little lymph vessels, those guys are going to adjust and they're going to enlarge and hopefully take over. If that doesn't happen, you're always wearing a sleeve on your arm, a compression sleeve, to minimize any edema. Okay, so cells in the immune system. The biggies are the lymphocytes. So these guys have to learn how to work. These guys have to learn how to do their job. And we'll get into that in immunity. So in other words, they have to adapt. They're not born with the ability to do their job. They have to learn how to do their job. The both of them are in charge of protecting against antigens. So anything that's not supposed to be there, anything that your body says, hey, you do not belong to me, I'm going to kill you. I kill you. So that includes mismatched red blood cells. If somebody gives you a blood transfusion, it's not your blood. That's a foreign body. So that's an antigen. T cells actually can do some killing. Some of them are the major helpers. So we're going to have helper T cells that are going to help everybody in the immune system. But then we actually have some that kill. And then we'll do that in the immune system. B cells don't actually kill. So again, both of these are lymphocytes. And when you looked on that slide in lab and you saw those different lymphocytes, you can't tell really which ones are T cells and which ones are B cells. But they're both part of the immune system. They both want to take care of antigens, but they work a little bit different. The B cells are the guys that when they get activated, they're going to release antibodies. So when they get activated, their name's going to change from just a plain old B lymphocyte to a plasma cell. And a plasma cell is the guy that's going to release antibodies into the bloodstream to attack foreign antigens. Some other cells besides the lymphocytes, macrophages, dendritic cells, both of those are phagocytes. The dendritic cells are going to be more from the epithelial tissues, and then macrophages are more from the connective tissues. When we get into the immune system, we'll talk about how they can function kind of differently. Both of them are going to eat up foreign bodies, but they're going to present that antigen to another cell, to a lymphocyte. So they're going to gobble, 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 and then they're going to carry it to a lymphocyte. Macrophages do a whole lot more gobbling. So most of the time when we talk about a dendritic cell, his main job is to gobble up one antigen and bring it and go tell the lymphocytes, look what I found. This is bad. Do something about this. Macrophages can actually do something about this, but they also will take it to the lymphocyte. So macrophages might be found very plentiful when you have a whole bunch of, let's say you got an infected cut. You could find a whole bunch of neutrophils eating the bacteria, and then we're going to have some macrophage come and clean up the mess and eat all the dead neutrophils and stuff like that. But they also can grab that antigen and take it to a lymphocyte. Dendritic cells gobble up one and go and tell, look what I found. Reticular cells are actually going to be found inside the lymph organs, and very similar to a fibroblast, they secrete the fibers that make up the lymphoid organs, and they give macrophages and dendritic cells kind of something to hang on to. So here's a picture. Let's see. All these little things coming like this. These are all reticular fibers, and if we look really hard, we can probably find a nucleus for a reticular cell. Perhaps here. Maybe maybe that's a nucleus. But anyway, just like the fibroblasts secrete the collagen and secrete the elastin fibers, the reticular cells secrete reticular fibers. And if you remember from AMP1, we kind of skipped over that because you're already learning enough about tissues and we get into it into AP2. Well, here you go. Here they are. And somewhere in here, there's going to be cells patrolling. There should be some lymphocytes and macrophages. Here we go. This is showing a macrophage up at the top. That's kind of hanging on to those reticular fibers, kind of giving him a place to just chill and watch as things pass through that lymphoid organ. And he's just patrolling. And then lymphocytes can do the same thing. Okay, so we have different kinds of lymphoid tissue. We have some of them are going to be scattered little patchy areas. They're going to be called diffuse. Some of them are more of a structure called nodular. And then that can be broken down. Some of those nodular guys have a capsule, some of them don't. But the main function of this lymphoid tissue is a place that's housing the lymphocytes. So give them a place to kind of hang out so they can first meet the antigen and they can get busy and start learning how to destroy it, so get activated. And then we've got macrophages living in there. Maybe we have some dendritic cells in there. Just things with lymphocytes hanging out. So like surveillance areas, just waiting to be activated. Those immune cells waiting to be activated. So primary lymphoid organs is where the cells, the B cells and the T cells, are actually going to mature. So they're all created in the bone marrow. You remember that. The red bone marrow makes red cells, white cells, platelets. Well, lymphocytes are a type of white cell. So that's where they're made. B cells can go ahead and get matured in the bone marrow. So B for bone marrow, B cells. They mature in the bone marrow. But the T cells actually have to leave the bone marrow and migrate to the thymus. So T, thymus, T cells, that's how they learn how to do their job. They actually go through a lot of testing. The thymus puts them through a lot of testing. So for all the lymphocytes that are actually sent to the thymus, the percentage that actually get to go out and leave and do their job is pretty small compared to the ones that get sent there. Okay, secondary lymphoid organs are the guys where those lymphocytes are going to be activated. So they're going to meet their antigen, and when they meet their antigen, they're going to create armies. Lymph nodes are the biggies as far as they're doing the majority of the screening, but we also have different areas where we have T cells and B cells that are hanging out and waiting to do their job too on surveillance. Lymph nodes are the major filterer of lymph. So here's some areas. Tonsils, we've talked about those guys before. Red bone marrow and thymus are primary, but tonsils are secondary. Spleen is the secondary one. Peyer's patches, you met those in the ileum and even the appendix. All secondary. So, the main dude is a lymph node. So the lymphatics, the lymphatic capillaries are going to be bringing eventually to bigger vessels, going to take it up to the lymph nodes. And usually it's going to be a whole chain, a lymph node chain. So it's going to go through one, and that lymph is going to get filtered for antigens, cancer, bacteria, dying cells, whatever. It's going to filter the lymph. Then it's going to leave that cell. We're going to filter it again. Leave that lymph node. Then and we're going to filter it again at the next lymph node. And then we're going to leave that lymph node and we're going to get filtered again. So hopefully by the time it's done, it's all cleaned up. The macrophages that are hanging out in there or the lymphocytes that are hanging out in there have done their job and made sure that that infection did not travel to the next area of the body. So again, you have that right lymphatic duct that's going to drain mostly the right side of the head, the right arm, and the right thorax, and then everything else is going to go into the thoracic duct. So we have the lymph passing through it and it's getting filtered. We don't want any of those unwanted things spreading to other parts of our body, including the blood, because that would be sepsis, yes? The other thing, those lymphocytes that are hanging in there, they're hanging out and they're waiting to be activated to create armies. Those lymph nodes can be very small, so they can be millimeters in width. Most of them look like beans, and the largest is less than 2.5 centimeters, so less than an inch. They have a capsule around them, lymph nodes do, and then they have these little connective tissue lines that are going to dip down into the lymph node and kind of separate it into separate areas. You remember when we say cortical bone, that is the outside hard bone, the medullary bone is the inside of the bone, and I've said it with hair and adrenal glands, kidneys. We haven't done that yet, but we will. The cortex is the outside, the medulla is the inside. So I'm going to show you a picture in just a second, but the cortical area has little germinal cells, centers that are full of blue ovals. And those germinal centers that look like blue staining ovals are full of B lymphocytes in the cortex. Surrounding those germinal centers, especially deeper in the lymph nodes, is where we're going to find the T lymphocytes. There's also going to be some dendritic cells, so the guys that are going to be presenting antigen to the lymphocytes. In the medullary area, we have all of them. We're going to have some B cells, T cells, plasma cells, and we're also going to find some macrophages in this deeper area, and just checking out the lymph as it's passing through. So here is a drawing of a lymph node. Notice here's an afferent, so A arriving, efferent, E exiting. Look, 1, 2, 3, 4, 5 afferents arriving. This is the cortex right here. This is the medullary area, so deeper towards the middle. These little areas are called the germinal centers, and they're loaded with B cells. Deeper in the cortex, we're going to find some T cells. This is the connective area, so this is the connective tissue capsule that's on the outside with these little dips that are going inward. Those are called trabeculae. And then down deep on the inside is the medullary area. You can find some B cells, plasma cells, T cells, macrophages, all doing their job. So what's the function of having so many lymphatics going in, afferent, but only a couple going out? This is just a histology slide, not a drawing, but it's the same idea. So this right here is a germinal center full of B cells, and then surrounding it we're going to have some T cells. Well, the purpose of that is, you can imagine if you're pumping in at many areas lymph, but there's only a couple of exits out, it's going to be a slower flow because there's only 2 exits. So, you know, at an arena, if you're at a concert or something, that would be very dangerous. So if something happens, like a fire breaks out on the stage, and there's only 2 exits, we're going to create a song called Smoke on the Water, because people can't get out when there's a fire on stage. It slows down the exiting. But why would we want to slow down the exiting? Well, if we're going to slow down the exiting, it allows more time for that lymph to be monitored, to be patrolled. Hey, are you okay? Are you okay? Hey, do you see anything bad over there? I think I might see something bad. Let's go and see over there if there's something bad. And if they see something bad, they're going to grab a hold of that antigen and gobble it up and present it to the lymphocyte. Here's an example of when the lymph nodes have been overwhelmed by some sort of antigen. So, if you can appreciate, in his armpit, he has a very large swelling. And the name for this swelling is called a buboes. That's where bubonic plague got its name, because that's very typical of bubonic plague. If you've ever known anybody with cat scratch fever, very same idea. They'll go in and lance that lymph node. Sometimes these guys will be misconstrued or sometimes called swollen glands. So if your lymph nodes in your neck get swollen, sometimes somebody might say you have a swollen gland. Well, that's inappropriate, actually, because a lymph node is not a gland. Now, if you had the mumps, that's swollen parotid glands, that's a swollen gland. But maybe you remember me talking about adenoids when we talked about tonsils. And so when something is oid, it's gland-like. And so just a quick quiz, do you remember which of the lymph nodes, when they say they're going to remove their tonsils and their adenoids? Adenoids are tonsils, and those guys would be pharyngeal. They're in the nasopharynx. So gland-like, but they're lymphoid tissue. I mentioned this before. So if the lymph node is draining a cancerous area, then what can happen is some of those cancer cells break free and get into the lymph nodes. They can keep on going. They can go through. If they don't get caught by a macrophage and gobbled up, then they can keep passing to the next node, into the next node, into the next node, and end up getting dumped into the subclavian vein. Now it's in the circulatory system, and it can spread anywhere, and that's called metastasis. When you have a swollen lymph node because of cancer, or maybe it's a primary lymphatic cancer, maybe it's started in the lymph nodes, whatever, the lymph node can get big, but it doesn't hurt. If it's infection like bacteria, it does hurt. Okay, spleen. This guy is very cool looking, and he's very simplistic, actually. So that splenic artery, if you remember, the celiac artery branches into the hepatic artery, the gastric artery, and the splenic artery. So here we go. That splenic artery is going to feed into the spleen at a hilum, and the lymph node had a hilum too. And it's going to first, where those little arteries are going into the spleen, those arteries are going to be surrounded by lymphocytes. So that is going to be called white pulp. And the reason is, when you look at a spleen, let's say you do a cross section, not on a stain, but if you look at just a spleen grossly, it's going to look like it has white dots in it. So everywhere where you have an arterial vessel passing in there, it's going to be surrounded by lymphocytes, so white pulp. All the rest of the spleen is red pulp. So what's going to happen is, as those vessels pass through, you're going to have some of those sinusoidal capillaries now. And if you remember, those are the guys with the biggest holes, the biggest gaps, like large Swiss cheese holes that things can pass through. These guys are going to be, the red pulp is essentially macrophages and red blood cells. So the purpose of lymph nodes is to cleanse lymph. The purpose of the spleen is to cleanse blood. So we're going to have macrophages that are cleaning up any sort of debris. Did we get some sort of parasite in here that's not supposed to be here? Infection like Mycobacterium. Hopefully, spleen's going to help gobble up any of those kind of bacteria because those guys are hard to combat. So maybe it made it into your bloodstream and your spleen sees it, but hopefully your red pulp is going to help catch it with all those macrophages in there. Also, red blood cells have a lifespan, approximately 4 months or less. So we're going to end up recycling the red cells and gobble them up, break them up, maybe recycle the iron, and send anything left through that splenic vein. That splenic vein is going to dump into the hepatic portal system. So it's all going to go to the liver. Another thing that the spleen does is he is going to be a place for platelets to hang out. About 30% of our platelets are always hanging out in the spleen. Oh, good, there is another slide. I was hoping there was another slide. Because then I'd be saying all this stuff and it's not written down anywhere. Okay, cool. So we're going to break down those red cells and we're going to save, especially the iron, save it for use. We're going to store some platelets, also store some monocytes, so that when there is a need for extra, it's going to release it. Oh, I forgot to mention this one. And the third thing is, as a fetus, a lot of our red blood cells are made in the spleen. When you're born, your bone marrow completely takes over of it. He has a fibrous capsule, although his capsule is kind of thin. And you probably know this because you've heard of somebody that's been in a car accident or maybe bull riding or bronc riding or something like that and broke their spleen and they bled. When we talk about breaking their spleens because they ripped that thin capsule. Said all this already, but the white pulp, we've got some lymphocytes that are surrounding those arteries as they're coming into the spleen, and then the red pulp is where we're going to have some macrophages to eat anything that's not supposed to be there and eat old dying red blood cells. Use that iron, send the rest of the stuff to the liver. See, here's the thing. When you stain something, you can't tell it's red and white anymore. You have to, it's when you look at it grossly, like I said earlier. You cut the spleen and you look into it, you can see little white dots. But when you stain it, those B lymphocytes are really going to pick up a blue, so you don't look white at all. But you can appreciate that these little centers look different. Wherever the white pulp is, histologically, it looks different from all the surrounding area. So here we go. Splenectomy. Bronch riding. Ruptured your spleen. Now it's full of blood, right? Lots of red pulp. Lots of red blood cells. So you're pooling blood into your belly. You can bleed to death without it ever leaving through your skin. It's just dumping into your abdomen. You can bleed to death that way too. Hypovolemic shock. So we used to always just take the spleen out. It's bleeding. Let's take it out. Now what they'll try and do is they'll give you whole blood and replace the blood that you're losing and give it time to see if maybe it will stop bleeding on its own so they can save that spleen. If you lose your spleen, your liver actually can take over, but it would be nice if you just, didn't lose your spleen if you didn't have to. Also, children, if you leave just, let's say that they bad accident, losing too much blood, you can't take a chance, so let's go in and do a splenectomy. If you can leave one little part of the spleen that hasn't been damaged, it can regenerate. So that's awfully cool. Okay, the term MALT, mucosally associated lymphoid tissue. So this is going to be lymphoid tissue that's going to be in a mucosal area. You have some diffuse MALT that you can't really see, but we're going to talk about 3 areas that are very evident. You've met them already. So tonsils, Peyer's patches, and appendix. So these are actually like clumped areas of mucosally associated lymphoid tissue instead of just diffusely passed through. Every organ has diffuse lymphoid tissue. So this is going to be a review. Tonsils are the simplest of all of our lymphoid organs. There's one that we hadn't mentioned before, the tubal tonsils, and I kind of thought about not even putting them in here again, but oh well. There they are. They're surrounding the auditory tubes as they're dumping into the pharynx. So trying to keep, when you eat, I think I told y'all my children all ate a cricket. What else was on that cricket? I don't know. But at least it didn't get up into their ears and they get an ear infection because they had a cricket. So these tubal tonsils are protecting because your eyes, ears, nose, throat are all connected. So we don't want anything that we put in our mouth to transcend up into our ears or higher. Palatine tonsils are the one that you can actually see when you open your mouth really wide. We've talked about this. They're the most commonly infected ones. These guys, why do we have these guys? We have these guys, they're like the first guys to meet many of the invaders of our body. And they can put a stop to things right in their tracks. So air, food, whatever. They don't have a capsule, but they have those germinal centers with some B cells and then surrounded by some T cells scattered around. There's going to be some macrophages in there. And what's going to happen is we're going to get the ball rolling with this for some of our lymphocytes to learn how to do their job. They're going to make armies and they're going to create some cells, not just armies, but cells called memory cells that we'll get into in the immune system that's going to say, hey, I've met this before. I can create an army really quick. Memory cells can make an army in a flash. But when we're first activating lymphocytes, the army building the first time is a little bit slower. But once you've created an army, some of them will hang out as memory cells so that the next time that antigen comes around, it can create an army lickety-split, like the reason you do second dosing on the vaccines. First time takes 10 to 14 days to get an immune response, the army. But the second time you get the vaccine, you get the army in 24 hours because of memory cells. Don't learn the histology of a tonsil, but look at the picture of where they are because that's a good test question. Look at those, Palatine. Blech, that's disgusting. That is strep throat. Okay, Peyer's patches. So these are lymphoid follicles that are in the ileum. Same thing. So they're in the epithelium, so the mucosal area of the intestines, and they're constantly killing bacteria and making sure that they don't sneak into the bloodstream. This is what they look like. All these little lots and lots of nodules, lots and lots of B lymphocyte areas. And then the appendix also, that's where the small intestine is meeting the large intestines, and that's full of lymphoid follicles. So some bacterial destroyers in there. Also going to create memory for the next time. Okay, so the thymus is actually a little bit different from other lymphoid organs because their function is only for T cells to mature. So a thymus is largest at like 1 year old. It grows a lot that first year, and then it slowly regresses. And you probably remember this, once we hit a certain age, 7, 8, 9, 10, then it gets a lot smaller. Not a test question as far as age, but what happens as we get older? It becomes a whole lot more fat and connective tissue and a lot smaller of a thymus. It still works. It's still maturing T lymphocytes, but as you can imagine, if we've got these long-lived cells in our body, we don't have to have that thymus working so hard as we get older because we've done a lot of the work already. Don't memorize the histology of a thymus. And this is a review of just a chart of what all these guys do.